I'm Helen Syrett, a member of the Beatrix Potter Society. The Beatrix Potter Society aims to celebrate the life of and works of Beatrix Potter and seeks to inform the public about and promote research into the life and work of Beatrix in charitable ways and for the public benefit. Today, July 28th, is Beatrix Potter's birthday. It's her 156th birthday, and we are delighted to join the Linnean Society in presenting this program on Beatrix Potter's mycological explorations. I want to extend a warm welcome to all the Beatrix Potter Society members who have joined us today, including our speaker, Dr. Fred Rhoades, and his wife, Gloria, as well as the members of the Linnean Society and all of our guests today. More information on the Beatrix Potter Society can be found at www.beatrixpottersociety.org. With that, I'll turn it over to Isabelle from the Linnean Society.
Thank you very much. And it's a pleasure to be here tonight. So I'm not going to spend too much time. I am going to share my screen. I have a presentation which is mostly about Beatrix Potter and the Linnean Society, because a lot has been written about this, and I just thought I would present you with the facts as we have them in our archives. So this is only going to take about 10 minutes, and then I'll pass over to Fred for the main talk. So the history and the story of Beatrix Potter and the Linnean Society really fits in with the history of women at the Linnean Society and other learned societies. Women were first admitted to the fellowship to be fellows of the Linnean Society from 1905. Just as a reminder, the Linnean Society was founded in 1788. And in terms of learned societies, this is seems quite late, but actually a lot of the learned societies welcomed women a little bit later than that, and up to after the Second World War in the case of the Royal Society. So before 1905, and before women became fellows, what could a woman who was interested in science and participating in the scientific life of the Linnean Society do? So a woman could attend a meeting, but she had to be accompanied by a male fellow. She could submit a paper, but it had to be sponsored by a male fellow. So while it wasn't impossible for a woman to get her scientific work recognized, read at a meeting of the society, and then published in the Linnean Society journals, it was difficult, and it required a lot of networking, knowing the right people. Beatrix Potter had access to some of these networks, so she knew the mycologist Charles McIntosh, who supplied fungal specimens to Potter. He also guided her painting. He recognized her wonderful draftsmanship. And her uncle, Sir Henry Roscoe, introduced her to someone who would go on to become another great Potter supporter, and we see him on the right, George Massey, keeper of lower cryptogams at the Royal Botanic Gardens in Kew. So the story of Potter and the Linnean Society is often related as the Linnean Society treated Beatrix Potter relatively unfairly, having refused to publish her paper on the grounds that she was a woman. And while it's undeniable that she was probably had to go through a lot more hardship than her male colleagues, the archives show slightly different versions of event, but you need to kind of navigate between several archive records to draw the full picture. And as we'll see, we don't have the full pictures because some of these records are missing. So if we look at the council minutes and a register of papers received at the society, they indicate that George Massey presented a paper by Beatrix Potter entitled On the Germination of the Spores of the Aragacinae on the 18th of March, 1897. And the council minutes in here are blown up the relevant section, indicate that it was ordered to be referred, and given the paper was given the number 2978. And then that paper was then listed in the register of paper that were read at the society, where you can see the number here, the fact that it was received on 18th of March. There's her name there, George Massey, who presented the paper, and the title of the paper, and the further indication that it was read at a meeting of the Society on the 1st of April 1897, but then how it was disposed of, whether it was declined or printed in a journal, is left blank. We have a further register of papers which was used to list papers which were submitted to be published after they were read at a meeting of the Society, which indicates that the referee was George Murray. George Murray was a Scottish naturalist who was closely associated with T.H. Huxley and with the Discovery Expedition. So the paper, as we see here, was sent to the referee, George Murray, on the 23rd of March, and he reviewed it within 2 days and sent it back on the 25th of March, 1897. And then the council decided on its fate on the 8th of April. So the paper, as was noted, sorry, my mouse has stopped moving, here we go, was read at a meeting of the Society on the 1st of April. And so we now turn to the general meeting minutes, which record every single meeting of the Society. And you can tell from the number present that it was read in front of quite a crowd, actually. It is rare that Linnean Society meetings would have had such a number of fellows attending. And you see here the name of George Massey, who was the one who read the paper. From the list of visitors, the name of visitors here, we see that Beatrix Potter was not actually present at that meeting. And then the minutes indicate that her paper was read and discussed on the germination of the spores of Aragicinae by Miss Helen B. Potter, communicated by Mr. George Massey, FLS. So it's not unusual. In fact, it was not usually the norm that authors would read their own paper. So it's not because she was a woman that she didn't get to read her paper. In fact, even the most famous paper at the Linnean Society, and I'm referring to Charles Darwin and Alfred Wallace's paper on the theory of evolution by natural selection, was not read by them and was read in their absence. So it wasn't, it was perfectly normal that someone else read the paper. Finally, the last archival record, or the council minutes of 8th of April, indicates that Beatrix Potter was asked to do some corrections, whether it's following the discussions or as recommended by the review. That's not known because neither are extant in the Society's archives. But we know that Beatrix, we know that she was asked for more work because she wrote to Charles Mackintosh, and her letter indicated, and I'm quoting, "My paper was read at the Linnean Society and well received according to Mr. Massey, but they say it requires more work in it before it is printed." And this last record indicates that for a reason that is unknown, she never did resubmit the manuscript, but instead she withdrew it. So we have here in the council minutes a proposal on behalf of Miss Helen Potter to withdraw her paper was sanctioned. And as a result, within the register of papers submitted, under remarks, it says withdrawn. As you can see from the list, it's not the only paper to have been withdrawn or declined as well within a short period of time. So in conclusion, I'd say that the process which followed Potter's submission of a paper to be read at the Linnean Society was pretty much usual for the time. But it seems that she, for one reason or another, never resubmitted the manuscript, and it was withdrawn probably at her request. It's difficult to get a full picture given that the manuscript, as well as the peer review, no longer exists, or at least we've never found them. And in fact, and as we were just saying before the meeting started, and as Fred was remarking, it's a great shame that this manuscript was not only never published, but also seems to have disappeared. And one can only hope that it will resurface one day to kind of, I think, honor her contribution to the discipline of mycology in the way that it should. So I just wanted to finish by saying it's linked to that, because we don't have that paper. It's a great shame as well, because we know that she encountered more hurdles in her scientific work because she was a woman, and that these hurdles probably deterred her from pursuing more research in the field of mycology. And this was a great loss to the field. There's no doubt that she was a consummate illustrator who closely observed fungi and faithfully recorded what she saw. And I think we're going to learn a bit more about this through Fred's talk. So I'm going to stop sharing and pass over to you, Fred. Thank you.
And I'm just going to intervene. I'm Marta McDowell, a member of the society, the Beatrix Potter Society, as is Dr. Fred Rhoades. And so I am pleased and honored to introduce him, as well as to discuss questions after his presentation. Dr. Fred Rhoades is a retired instructor and current research associate at Western Washington University in Bellingham, Washington, where he photographs cryptogams and inventories lichens and mushrooms. He and his wife, Gloria, are members of the Beatrix Potter Society. Welcome, Fred.
Well, thank you very much, everyone, for having me. I need to figure out how to run this here. There we go. Actually, when I was hired at Western, I was hired as the cryptogamic botanist, which included fungi and other cryptogams, not cryptograms. Cryptogams, the spore-producing plant-like organisms. I thought I would review some of the things that Beatrix Potter knew about the fungi and other cryptogams, and basically celebrate her wonderful, wonderful artwork in illustrating these, but also talk about the experiments that she was doing with some of them. I'm very fortunate that I was able to get quite a few illustrations. Particularly want to thank Sue Osman of the Armitt Trust and Anne-Marie Bilklow of Victoria and Albert Museum, who supplied me with some images, some high-res scans. But a lot of what you will see, I've purloined from a variety of places. I've taken the liberty to touch up some of the photographs color-wise, brighten them up, and otherwise have not done a whole lot, but I apologize if anybody feels insulted in the results you see. Not all are that wonderful, but anyway. What I plan to do, eventually get to the fungi, but I want to talk a little bit about some of the other cryptogams that she illustrated, and overall talk about what was known back then in the late 1800s and what we now know about some of the things that Beatrix opined about. I recommend these resources if you're just getting into this area, particularly a Victorian naturalist, which has wonderful chapters by Mary Noble and Anne Stevenson Hobbs on first the mycology aspect of her drawings, and Stevenson Hobbs looks at the art, and both those chapters cover way more than I can talk about, but very, very well done. Linda Lear's biography, and of course the translation of Beatrix Potter's secret journals, very interesting reading. If you want to see more of her illustrations of fungi, Finley's early guide and this more recent guide in French that Anne Stevenson Hobbs edited are sources of the illustrations, as are these things up here. And then Roy Watling, a Scottish mycologist and Mary Noble, also a Scottish mycologist, have written some very detailed articles tracing Beatrix's work on fungi, both well worth reading for more details, more than I can get into here. One thing I thought I'd start out with was talking a little bit about what was understood about the evolutionary relationships of all the organisms that Beatrix studied and drew. And back then, of course, there were plants and animals, and Ernst Haeckel, a German biologist who had a very modern view back then, and many of you are probably familiar with many of his fantastic drawings of all kinds of life forms. But he had this wonderful tree of life. You have the plants and the animals, and then he, unlike many people, realized that there was this whole group of little things, the protists, and noticed that the fungi and the lichens are treated as plants. Other things that Beatrix drew pictures of, the red algae, the brown algae, the mosses and other bryophytes, and the slime molds are distributed on this tree, as you see. In 1963, Robert Whitaker, an ecologist, came up with the 5 kingdom system. The main thing here, and this is now out of date, I'll show you in the next slide. The main thing is that the fungi were regarded as a separate kingdom, largely because of their ecology, but he placed them stemming from the protists, the simple organisms with nuclei here, and of course everything came from the monomer. Today's view is much more complex, and by looking at molecules and comparing different molecules across all organisms, this viewpoint, this is just one of many, this is a particularly nice one I like. Notice that the eukaryotes, those things with very complex cells, have a huge amount of evolutionary lines. The animals are on one line, the plants are on a completely different line, and strangely, well, maybe not so strangely, the fungi are actually very closely related to the animals. Bacteria here and another prokaryotic group. And then all the things that, the other things that we're going to talk about are distributed across the board in very many different lines of evolution. Things in brown here are what had been classified earlier on in this century as fungi. Now we realize that fungi is a polyphyletic group. Well, I'm going to talk about a few of the other cryptogams first, and then we'll get into the fungi, including the lichens. As far as I can find, there are virtually no what one might call scientific illustrations of ferns. This is a beautiful illustration of fiddleheads, which I think are a bracken fern. I love this quote that refers to the fact that Beatrix wasn't formally educated. She had governesses that basically allowed her to do the things on her own that she did. And she kept her cool and wasn't swayed by everybody else, and was able to go on and look at things rather independently. Later, this is much later illustration. Here's some pictures of ferns, but mainly an artistic rendering. Algae, the lower plants, they're divided into the red, green, and brown algae. As far as I know, this is the only image of a red alga that Beatrix Potter drew. There may be others. I just haven't been able to get to all these images and look at them closely. I think this is what's now known as the sea comb seaweed. a beautiful little marine alga, Plocamium. I'm not positive about that, but I'm pretty sure. In this montage, early still life, we see a seaweed, probably rockweed, fucus. By the way, in this talk, I'm mostly using the scientific names, either the genus or the species names, which are binomials. I know some of the common names, but common names between here and there differ, and I'm much more convenient with the scientific names, so that's what I'll use. Next, I want to turn to the bryophytes. The bryophytes include the mosses, the liverworts, and the hornworts. When Gloria and I were on the Beatrix Potter Society tour in 2014, we were very nicely shown some of her bryophyte illustrations upstairs in the Armitt, and I was taken by the beauty of these. Bryophytes are beautiful organisms, particularly up close. The taxonomy is based on, ultimately on, let me talk a little bit about the biology of bryophytes if you don't know about how they're built. Bryophytes, like all plants, have two phases. A gamete producing phase called the gametophyte, that's the green leafy plant that you see, and a spore producing plant, which is a separate phase of life, called the sporophyte. Sporophytes aren't always present, even though they do kind of form the basis of the overall taxonomy of the group. So features of the gametophyte are very important. Things like cross sections of leaves, which as far as I have seen, Beatrix Potter never illustrated. Now there may be some, and I'd love to see them. But surfaces of leaves, the presence of midribs, dentation along the leaves are all important. Branching patterns of the gametophyte. Features of the sporophyte are interesting if you have them. And I know there's an illustration in the Victorian Naturalist, which is improperly labeled. I want to just point out that the capsule, the spore producing capsule, has a lid. This is all sporophyte, which this breaks off, exposing teeth. We'll see those in the next picture that spread the spores. But often on top of all this is a hood, the calyptra. This is actually leftover part of the gametophyte that has grown up with the sporophyte. It's the part that housed the egg that was fertilized early on to form the sporophyte. If you're interested at all in all this stuff, I refer you to a biology book. You can study the life history of mosses and other bryophytes more than I can talk about it. Just a few other illustrations showing her command of macro illustration. All this would have to have been done under a hand lens. Here you see the sporophyte with the teeth exposed. There, the lid and the calyptra, the hood. Here, the leaf with showing a central midrib and the dentate margin. The overall habit of the way that these grow in tufts is important. It's a shame. Apparently, there are. Here's that. Apparently, there are some 28 moss illustrations in the Armitt, and they're lovely, lovely drawings, often illustrating at extreme magnification features that would be useful in identifying the species. And it's a shame she didn't do more. They could be used to illustrate a book, I would think. Another group of cryptogams are the liverworts. And as far as I have seen, she illustrated two. One, this thallose liverwort, thallus liverworts have strap-like gametophytes. This is Marcantia polymorpha. It's a common greenhouse and garden weed. I always like to see it in my garden. Anyway, everything you see here is female gametophyte, and the sporophytes form when the eggs formed up here are fertilized. What she's illustrated at very high magnification here are the surrounding leaf-like structures around where the egg and then sporophyte would form. And then she also includes a magnified view of the pores. This is a very complex plant that has little air pockets with pores in them, much more advanced than many liverworts. Not sure what this is. It's faded so much it's not real clear what is being illustrated here. Perhaps details of cells inside the chamber, not sure. I'm throwing in just an illustration because I love this plant so much. And Gloria and I were looking at plants at a local nursery last week, and there, lo and behold, was this lovely little display of Marcantia. And one of the things you can see in this picture are the female structures that bear the eggs. But the thing that Beatrix didn't know about was that there are male structures that bear sperm. They're little on separate plants. This has separate male and female plants. These are the male structures here that she did not illustrate as far as I know. And then the other thing you see here in abundance are an asexual stage. These are called gemmicups. They produce little buds that splash out. And this is why this is such a noxious weed as far as gardeners are concerned. But like I say, I always like to see it. Another group of liverworts are the leafy liverworts. These are less well known. They look like mosses. They're often called the scale mosses. Here's one illustration of a leafy liverwort that Beatrix did with a good close-up. And the arrangement of leaves and the shape of leaves overall on the plant, very important feature. Not sure what this is here, but another magnified view. Moving to the fungi, it's interesting that in her little books, there's only a single image of mushrooms or fungi. Of course, she was doing these books in the early 1900s. The tail of squirrel nutkin includes this tiny tuft of mushrooms. Maybe that's Armillaria mellia. I don't know. It's not detailed enough to know what it is, but that's the single illustration of mushrooms in one of her fantastical little books. Another later illustration in a book, my understanding is it never got published, shows the Toad's Tea Party. Here are the toads on the toadstools at this tea party. I don't know if this was ever pointed out by anybody, but that's a wonderful drawing. Apparently being served a cake with jelly, witch's butter jelly fungus, but that's not clear here. Anyway, that's another one of her fantastical drawings, including fungi. But mostly she didn't include fungi in her drawing, her whimsical drawings, even though she knew very much about them. I'd like to just review a little bit about the features of the fungi for those of you who are mycologists, talk a little bit about their biology for a second, then we'll look at illustrations she did of lichens and the larger fungi. Here's a cone with some mushrooms on it. Mushrooms and other fungi are the visual occurrences of a much more diffuse body called the mycelium. You can see it here amongst the scales of this cone. The mycelium itself is composed of smaller units called hyphae. They're little tiny tubes. And it's the mycelium that does the ecological work of the mushroom, and then later builds with some of its hyphae these macroscopic structures that we call mushrooms. And of course, the whole idea of a mushroom is to produce spores. Often there are features of the mushroom that help produce lots of spores. There are two kinds of larger fungal organisms, two basically classes of, or phyla of fungi. I just wanted to review the differences for those of you who are not aware. And I'm just using this illustration, which we'll come back to at the end and talk about, which shows spore germination. But she was actually looking at this fungus, which is a basidiomycete. The basidiomycota form their spores on a basidium, a club-shaped cell, usually has 4 horns on it, with spores formed externally to these. Then they usually the fruiting bodies here, these look like little cups. And Beatrix thought this was a lichen at first. Not sure she ever really knew its actual existence, but they form these little cups that shed the spores from the basidia. In contrast to the basidiomycota, the ascomycota, which may also be cup-shaped, most of the basidiomycota aren't cup-shaped, but the ascomycota form their spores in asci, or singular ascus, usually 8 spores. It varies from group to group. Interesting that this particular cup fungus, which is now called Ramsbottomia, after John Ramsbottom, was an English mycologist, has these spherical spores that are very rough, look very similar to the Alurodiscus that we saw that was a basidiomycete. These microscopic details are often important in understanding what things are, particularly at the species level. A little bit about fungal ecology, which plays a part in our understanding of what Beatrix was doing. It's the mycelium that interacts and does the work of the fungus. And these jobs that fungi do fall into 3 ecological roles: decomposition of dead plants, a symbiotic union between the mycelium and the roots of plants called mycorrhizae, particularly important in the higher mushrooms, the basidiomycota and some of the ascomycota, in the pine, birch, and oak families. Other plants have other kinds of mycorrhizae. And parasitism, where a living host is attacked and often killed, or at least made sick, as true of both plants and animals. Actually, in the larger mushrooms, these first two roles are much more important, and we'll get back to that. Mycelia are often perennial, living from year to year, and due to environmental cues, put up their mushrooms at a certain time of the year. Temperature and moisture are often important, but other features of the environment play a role in whether something produces mushrooms or not. Something that Beatrix Potter didn't know about, and it was just becoming understood in the late 1800s and into the early 1900s, was the life history of a mushroom. And this is true for the basidiomycota in general. Ascomycota do things a slightly different way. But spores germinate if they're in the right environment, and they start producing enzymes that interact with the environment. If they're in the right environment, they get the food back into their spore and into their developing hyphae that cause them to grow further into a mycelium. Mushrooms spores start as what's called a primary mycelium, but in order to actually form fruiting bodies, they have to mate with another similar mycelium. And that's what's happening here, the fusion of two primary mycelia that have grown. Of course, this is all magnified. Then a secondary mycelium occurs. This has nuclei from both the original. They don't actually fuse at this stage. They remain separate. That's an interesting story. And again, I refer you to a biology text to get into more of the details of that. Ultimately, this secondary mycelium produces the mushrooms. So for a moment, let's not worry about the mushrooms and talk about lichens. I'm curious. I mean, a lot has been said about her feelings about the nature of lichens. And I think she was a very interested observer of lichens and curious about their true nature. But was she that interested? She didn't, as far as I can tell, illustrate in a scientific way many lichens. She was interested in spore germination. Here we are at Dalglish, where there are plenty of lichens to study if you want to draw them. But I am not aware of many illustrations of the kinds of things you see here. What are lichens? We now know that, like Schwendender, a German biologist in the mid 1800s, I think 1866, he proposed that they were dual organisms composed of more than one thing living together. Exactly what this living together, symbiosis, involves. Is it mutualistic? That is it a happy relationship for the two? Some people look at it as a controlled parasitism. I kind of like the mutualistic story. Obviously, they don't know what they're doing. They just find that it works for them. But lichens are formed from a fungus, which gives the lichen its scientific name, and often produce sexual fruiting bodies, and an alga, or sometimes more than one alga, actually an alga and a bacterium, a blue-green bacterium. It's called the photobiont. As far as we know, there's no sexual reproduction of the algae. They just divide by division. It's now known there are other fungi and lichens. It's getting to be a very complex situation. So they're really like little communities. Trevor Goward, a lichenologist up in British Columbia, has written some very interesting things about this. Look up Trevor Goward and read some of his ideas about lichens as ecosystems. Anyway, in the simplistic way of looking at growth forms of lichens, there are leaf-like ones, foliose, shrubby ones, fruticose, and paint smear ones, crustose. Here is a cross-section of a foliose lichen showing that the fungal partner forms an inner loose layer and outer surfaces, can't see the lower surface. And then usually just below the outer surface is a layer of the photobiont, either blue-green or green, as in this case. These are what Beatrix Potter and others of her time called gonidia. And she, I think, understood that there were different theories of growth, of the relationship of these organisms or these structures. And she tried to cultivate the gonidia and the fungus from spores. We'll look at a few of her pictures. Often they're very kind of fuzzy renditions. I'm not sure what the date of this is. might be early on, and I'm not sure if this is either a jelly fungus or a slime mold. In any case, that's not very descriptive. We'll move on from that. Here's a little more descriptive illustration of a fruticose lichen, stereocolon, which has lobes that can be more extended with lumps on the outside. Here's a local species, might be the same one as this. There are a lot of these stereocolons. They're a difficult genus to get species on, but you can see she certainly got the color right. And I must say that one feature of Beatrix Potter's illustrations across the board is the wonderful control she had over color. She gets the colors right every time. And that's no mean feat with watercolor. Anyway, this is one lichen illustration. The cladonias are a very common group. She had them listed under species which I don't think exist anymore, or at least aren't the right ones. I think this may be Cladonia bellidiflora, beautiful small cladonia seen growing with some mosses. I think she was mostly illustrated because she was drawing these at a time when she was really into germinating spores of fungi. And she was mostly interested because she had the technique of growing parts of lichens. From another species of cladonia, this is not, she listed it, or it's listed in the inventories as either of these two species, Cladonia filiformis or Pyxidata. Is definitely not Pyxidata, which has cups, and it doesn't have red fruiting bodies. So it's some other species. In any case, what we see here are results of her microscopic excising of the parts of this. And she gets the gonidia growing here, the algae, and she gets the spores growing. And most interestingly, this is the only cross-section I've seen of a lichen. This is of the fruiting body showing the asci. And you notice that one ascus, there's really just a sketch, has spores in it. The others are empty. This is true of lichens, and this is why she had so much trouble getting spores out of lichens, is they just don't produce a lot of spores. They're long-lasting thalli that spit out a spore every once in a while. If you do a cross-section, this is what you see. Mostly the asci are immature, and occasionally there's one with spores in it. So she rightfully had difficulty getting those spores out. And these are, in fact, cladonia spores. This is the shape of most cladonia spores, little oblong things that are germinating. So she was able to germinate them. I should say that now in the 20th century, the components of lichens have been separated and grown for long periods of time separately as cultures, and then put back together to form lichens again. This is an extremely difficult thing to do. It's very difficult to grow lichens in culture because they basically require alternating characteristics of the environment, wet, dry, and it's difficult to reproduce that in a lab setting. Anyway, here, this is actually just a few days after she did the previous illustration. She apparently was experimenting, looking at over time. Here we have January 13th through the 17th, a group of the algae. And this little group here, and this little group down here. And what I see is that whatever experimental setup she had to look at this, maybe growing on a Petri plate or in a hanging drop preparation, there's no indication of any growth of these. So I'm not sure that they were growing. Listed in Mary Noble's treatment in the Victorian Naturalist as part of the same study are these things. I don't know what they are. She says they're spore of the fungal partner. I don't think so. I'd be curious if anybody really understands what these are. Maybe cross sections of phalli. I don't know. Other pictures of lichens. I think this is not labeled lichen, but I believe these would be the algal cells and fungal hyphae from a lichen. And definitely this is a Pertusaria, a lichen where she is successful in growing over time the algal symbiont, what she was calling gonidia. Like I say, I think she was aware of Wendender's theory and was really maybe in partly these experiments were in her mind trying to get evidence that would support this theory. One final thing I should say before we leave the lichens, and this is a nice transition to the other fungi. There is a lichen that forms mushrooms. There are several lichens that form fruiting bodies of the Basidiomycota. This is a beautiful little species. Here seen very early on, really before she was doing much with mushrooms, just after she started doing mushrooms. This is a beautiful species. I don't think she really appreciated that it was a lichen. Here's my own picture of Lichenomphalia. Now, this is what it's now called, umbellifera, growing on a log in Bellingham here. It's a beautiful little thing. And the thallus is this green scum. And if you look at it up real close, you see little clusters of algae. And if you examine those microscopically, they're all enveloped with fungal hyphae that ultimately build these mushrooms. And this is where this mushroom gets its food. Doesn't decompose the wood, it is fed by its algae. So it is a lichen. Totally unrelated to the other lichens we've been looking at, which are largely Ascomycota. But the lichenized habit is one that has, it turned out, has evolved several times, many times in the Ascomycota, a couple of times in the Basidiomycota. Beautiful, it's a beautiful species. Turning to the larger mushrooms, I just wanted to review that there are lots of different kinds, and Beatrix Potter pretty much illustrated all these sorts of things, with the exception, I don't think she illustrated truffles or false truffles, underground fungi that she may have been aware of, but she didn't illustrate them that I am aware of. But we'll see illustrations of a variety of these things. I just want to basically give a kind of chronological look through her illustrations and talk about what features are seen in the illustrations, how well she was able to illustrate them in very naturalistic ways. Her illustrations, if you look, for instance, if you look at Finley's Wayside Fungi book, there are other illustrations other than Beatrix Potter's, mostly her illustrations, but there are others by other artists. And there's no comparison to the accuracy in realism of hers versus those other illustrations. What kinds of features do people look for in mushrooms? My list here is sort of focused on the guild species, to a lesser extent, other groups of mushrooms use some of these kinds of things. Habitat, where they're growing, what organisms they're growing with, and habit, the way they grow and how they're distributed in the environment they grow in, are very important features. And because Beatrix Potter did include habitat very often, some people complain that they're less scientific, but on the other hand, they include that additional information, which is important. And not all her illustrations include habitat, as we'll see, but those that do, I think it's an important addition to the scientific information in the illustration. The overall size and shape, very important in gill mushroom identification, is how the gills are attached to the stem up here. And as she went on, she learned that showing a section like this was a good way to illustrate that feature. Spore color is important. Usually the color of the gills is a clue, but there are other clues. I'll mention a couple. Presence of veils, none seen here. I'll talk about those in just a second. How the mushroom develops over time. This illustration very nicely shows that the young mushrooms have the cap bent around the stem, whereas the older ones, it flares out in a cone-shaped cap. By the way, the habitat in some books is illustrated on rock. I think this is actually wood. Sathirellas wouldn't form on rock, but they often decompose wood. Staining and microscopic details. Smell and taste are important. Obviously, you can't indicate smell and taste in a illustration, so you don't really have to worry too much about that, but they are certainly important features that, particularly at the species level, are useful for identifying things. Her early illustrations were still lifes. This very nice illustration of Amanita muscaria, the fly agaric, so-called, includes other life forms, Polypodium fern. I think this is the Thuidium moss. Here is lichen, barely understandable what this is, but it does give the impression of being Peltigera canina, and some English ivy there, and beech leaves. This can be, all amanitas are mycorrhizal. This could grow with beech trees, so that's a useful piece of information there. In the Armitt Trust, there are over 450 images of fungi. I was nicely allowed to see a few close up, but really, if you want to really understand what Beatrix Potter was illustrating, you have to see these, all of them, particularly her spore germination studies, which we'll get to. Here's a later illustration of the same thing, showing again the section which shows that the gills are free in Amanita, has white spores. Not so much on habitat here, but the veils show up very nicely. And we'll see more of these. The ring is formed by a partial veil that covers the gills during development, and the so-called the vulva is collars or sacs at the base of the stem, which in this species also part of that universal veil ends up on the cap. Colors, again, are very true to life. Oh, by the way, that was from the frontispiece. I'm not sure when that was done. Finley's frontispiece. Tooth fungus, now put in the genus Dentinum, Dentinum rapandum, the spreading hedgehog, so-called. Somewhat impressionistic illustration here. No habitat, of course, but you do get the structure of their often forms in clusters. These are mycorrhizal. Interesting, when she began working with Charlie McIntosh, which I'll talk about in just a minute, she did, and this was actually before then, but she did two illustrations of things. One which ended up in the Armitt. This is the version that ended up in the Armitt, and another that ended up in the Perth. Here I show that with this Helvella crispa, which does show some habitat, very nicely colored. This is the Armitt version, and here's the Perth version of the same thing. So later on, I guess I'm going to talk about Charlie in a second here. One other early, I think, or maybe very, very late illustration, which is again very impressionistic, doesn't really include enough detail to understand what this is this jelly fungus, now put in pseudoidenum. Here's my picture of it. This is a very attractive white jelly with teeth. It's not related to the other teeth fungi. Yeah, Charlie McIntosh was a Perthshire mailman who was an extremely good naturalist, and Coates wrote a wonderful biography of him, which traced his life. Says very little, I think because of the insistence of Beatrix Potter when this was printed, that she not be mentioned in his book. She'd moved on to doing other things and didn't want the publicity. But in 1892, she showed Charlie, finally got to meet him, having known about his interest in fungi, showed him her drawings, and he began to communicate with her, and from Scotland sent specimens to her in London, where she would draw them, paint them, and then send the illustrations back, at least one of them back to Charlie. He'd give his comments, and over time she developed a much keener eye. And really, from the following year, I mean, here's a very early illustration done probably the fall right after she first met Charlie McIntosh. This is a winter mushroom. Nicely shows gill attachment. You can't really discern that this has white spores, but habitat on wood, and the fact that gills are slightly attached, you can almost see that, but this illustration doesn't have a section per se. Interestingly, about this particular fungus, Flammuline evolutipes, I think you guys call it the velvet shank. It does have a fuzzy base to it, not seen in that previous illustration. But later, she had a stick that had this on, and she stored it in the dark, and another fungus showed up, shown here. And it turns out, she thought it was a completely different fungus, but it turns out it's the etiolated form of Flammuline evolutipes, which is now produced in culture and sold as a gnocchi, a Japanese mushroom. First, I think, developed this etiolation, growing in the dark in Japan. Anyway, she showed it, didn't quite understand what it was all about at a later point. So let's look at a few of her illustrations following her understanding, her beginning understanding of what mushrooms were. And really during this time period, 1893 through the end of the 1890s, when she got interested in spore germination, she really became a very good mycologist, learned the names, communicated with Charlie, and her illustrations begin to show her developing sense of what you should include in a scientific illustration. Although no habitat is seen in this illustration of Amanita rubescens, it does show a complete range of development. It shows the presence and appearance of the veils and the vulva of this species of Amanita. The gill attachment in this illustration is not really very clear. She does faithfully, as she does in all her drawings, include little bit marks, imperfections that make these so much more real. This is a favorite illustration of mine of bitter Boletus calipus, with beautiful rendition of color and an extraordinary feeling for depth that you just don't see in other illustrations. Not much on habitat here or other features that you might recognize in this bolete. The boletes are the fleshy pored mushrooms. They're Basidiomycota, and they're actually very closely related to the gilled mushrooms. Another bolete. This interesting species has been discussed quite a bit. I think Roy Watling discusses the history of her finding this in Dunkeld, and it turns out it was one of the first, I think the second sighting of this in Scotland. Interesting, known as the old man of the woods. Again, you don't see much in terms of habitat here. Certainly the color, the habit of growing in tufts sometimes, and aspects of the texture of the cap are well illustrated. The polypores are the fleshy, leathery and woody fungi. And little comment here by Mary Noble that by 1893, she already really knew the mushrooms extremely well. Here's an interesting mushroom in which you can see several features useful in its identification. It has a veil, a slime veil, which when that breaks up and leaves a ring on the stem, pulls some of the spores that have fallen on it against the stem. So you see this dark region, which gives an indication of the spore color on the veil remnants, and the gill attachments running down the stem are seen here. She, at the same time, in a separate illustration, did these, showing the veil, this cross section, showing the veil, the color at the base of the stem, and the development over time of this species. Very useful information. This illustration I threw in just because it shows how she approached the drawing of these. She drew first in pencil and then later filled in with the color. Another bolete, this one doesn't show a whole lot on habitat, but does show the fact that when you split this bolete open, it stains blue. Very important feature of what's now known as Xerocomelus chrysanteron. And this species of Lepiota shows the introduction of cross sections, which shows very clearly that the gills are free here, a feature of the genus Lepiota, along with the veil. Nothing on habitat here. This species is a brown spored mushroom. Often, Mary Noble has it upside down in the Victorian Naturalist. Mushrooms always orient their gill tissue downwards, so the spores can fall out. Never would they be oriented upwards unless the substrate was turned over. here is growing on wood, as Crepidotus do. It has brown spores, and there's a hint of something you see in nature that in a shelving species, it has a habitat of shelving like this, under the gills on neighboring caps, you'll often see a deposit of the spores, which I think is shown here. A jelly fungus, very true to form and color, with a lichen in the background. But notice that the lichen isn't nearly as closely observed. This is interesting to me. Getting close to the end of the fungi, I just have a brief few comments on spore germination in a second. One interesting fungus she illustrated is dead man's fingers, Xylaria polymorpha. This is an ascomycete. The asci are formed in these little pockets around the outside, which she illustrates very nicely in this cross section. When we were in Dunkeld, that was one of the fungi that I did see there. Here it is growing on a log. And the interesting thing that's seen here, and something that Beatrix Potter talked about, I don't know that she specifically talked about it here. This version of these fruiting bodies is not forming asci at this point. It's forming asexual spores in this gray coating. There was the Toulaine brothers, a French pair. One was a mycologist, the other an extremely good draftsman, put together a series of illustrations that illustrated that fungi do have many forms of spores. And they were particularly illustrating Ascomycota. And here is the view in their Selecta Fungorum Carpologica of Xylaria polymorpha, the dead man's fingers. Here you see the cross section, you see the little chambers, and these are the asci with the spores. Spores germinating here. This was done in 1863, so Beatrix Potter wasn't the first person to germinate spores. Other people had definitely done this. I think she maybe was one of the early people to work with mushroom spores. Here are the hyphae on the outside of the asexual stage, producing spores just by cell division. There's no sex involved in these. And this is something that is very common in the Ascomycota, something that Beatrix Potter alluded to in her thoughts, and thought that perhaps the Basidiomycota also did it, and in fact they do, although it's much less common and much less observed, maybe more common than people think, to have an asexual stage in addition to a sexual stage. Other fungi that she illustrated, some puffballs. This one, Lycoperdon, has a sterile base and then the spore sac at the top. She illustrated that. Cup fungi, again, here seen with lichens, an impressionistic view of the lichen in the background. The scarlet cup fungus always grows early in the year here, and I imagine that's true in England and Scotland as well, and inhabits small twigs, which are shown here. This is one of her final illustrations showing this species. And as she moved into the 1900s, away from her interest in mushrooms and into her little books, her mushroom illustrations, those that exist, are much more illustrations from an artistic point of view. She does have a cross section here of this species. Beatrix Potter was a wonderful observer of nature and very accurate recorder of the details of nature. And she got interested in the late 1800s in looking at things in a more scientific way, particularly spore germination of the mushrooms. Spore germination of mushrooms is fraught with problems, and that's illustrated here with this. We've already looked at this illustration. It's a basidiomycote, has little cups that orient downwards on sticks, produces spores that look like this on basidia, and here she got them to germinate. I'm not quite sure what process she used, but also in the same drawing are some other spores here and here. So there's a good illustration of an Alurodiscus spore. They're spherical and spiny, just as she is illustrated. Now, according to Watling, another local mycologist here, Jim Gins, had seen, I think here, a jelly fungus, maybe these little things, that's actually a parasite on Alurodiscus, Theotremella. Not sure if that's what they saw, but I'm wondering if these aren't the jelly fungus spores germinating. Here's another illustration a little later of what I think are Alurodiscus spores. But back to this illustration. This shows some of the problems with her technique. Wonderful that she was able to do as much as she could in her home lab, but they're always contaminants. And these spores are a mold, which here are the Tulane's illustration of this mold, alternaria, which is the asexual state of a little jug fungus called Pleospora. And you can see in the background here some of these alternaria spores germinating. Here's one here. Actually, the sexual stage looks a little different. It's different kind of spores. So these are asexual spores. So mold spores are going to be a common problem in many spore germination studies anyone does. She was successful in germinating spores. Often she got lovely illustrations of molds. Oops. The common mold Penicillium, it's virtually impossible. I don't know what technique she used, but it's virtually impossible to get a Penicillium mold illustrated with the spores and chains on the end of its little fruiting structure here. I'm not sure how she did this. It must have been in a petri dish culture of this mold. And finally, she presented her paper. And Isabella, I thank you for going through all the details of that, which I had no way of knowing, but it supposedly featured germination of the spores of Flammulina volutipes. In any case, I like this final picture of Beatrix and her brother Bertram. This was in 18, says there, I think, it's hidden on my screen, in the late 1890s, exactly the time she was doing all this fantastic work with mushrooms. What a wonderful person she was and how skilled she was. Well, I'll stop sharing and thank you for your attention.
Thank you very much, Fred. And what a wonderful way to celebrate Beatrix Potter's birthday in this joint meeting of the Beatrix Potter Society and the Linnean Society. So I just wanted to cover a few questions that I had and questions that have come up in the Q&A. So one that I'm curious about, and Margaret Payne asked as well, touched on, is I've always been curious about germination. And you said, you're not sure about how she did it. Do you kind of envision that she did this at home, at her home in Bolton Gardens in London, or, any speculation on that?
I believe these were all done at home, where it would be impossible to fully control contamination. If she did it in petri dishes, she could, use sterile media and then introduce the spores to the surface and then keep them covered when she wasn't actually looking at them. And then periodically observe the surface of the agar with the microscope. This can be done, but without a cover slip, without a proper mount, be very difficult to get clear images of things, which she seemed to get in her illustrations. Another approach to this, and maybe somebody who's listening knows more the technique she used, but another approach is called hanging drop, where you have a little chamber with a medium on it in which you grow the things, and then you can look down through the chamber and get a better view. I'm not sure which technique she used, but it was done at home.
Thank you. We had another question from Graham Rowe about, is there anything known about the optical aids that Beatrix used for the detailed aspects of fungal work?
Well, she had a microscope, and she started, as I understand it, with her, I'm not sure it was a gift or just borrowed, her brother's microscope, but then she maybe took that on for her own and acquired over time several different lenses. The highest magnification she was able to get with this microscope would be about 600 times, which is indicated on many of her drawings as 600. So she was using a compound microscope. I don't know if she had a hand lens to work with or a dissecting microscope to do lower magnification drawings, for instance, of the moss details that I showed that she drew. It seems that somewhere I read she had extremely good eyesight, so perhaps she was able to do the moss drawings with actually seeing those things with her eyes. But I think she probably had a small lens, a loupe, to use, as all bryologists and lichenologists carry a field lens to look at these features, and I imagine she had that. But for the spore germination, she had a compound microscope similar to the one that you saw in the illustration of Charlie McIntosh.
Thank you. You mentioned that, she would sometimes put like 100x on her drawings. And Margaret Payne also wondered, what was the level of detail that you observed in her labeling of her scientific illustration?
Well, how big things are is always tricky because there's magnification due to the screen you happen to be looking at and all the various techniques to get that image to you. So when you see times 600 on the surface of your computer screen, that really is not true. The actual drawing she did, and she was a very accurate, she drew what she saw. So I think on the sheet of paper that is the actual illustration, times 600 would be what she was seeing in her eyeball that she transcribed to the paper at the time. So she, using the, I was looking up because the quality of the lenses she used was indicated in some places. I think in her journal she got I think a 1/8 inch lens, for instance. I was looking up what you could achieve with various lenses that have those kinds of measurements, and the best that you could achieve with I don't have my little chart that I put together, but it was not 600 times. So she must have gotten a better lens with a shorter focal length to make those. Maybe someone has some details about the equipment she had. I'm not aware of what the actual details of the final microscope she was using.
Nor am I. One quote that I read, she wrote to a young cousin named Walter Gaddam. He was about 10 years old. And she wrote, "I have been drawing funguses very hard. I think someday they will be put in a book, but it will be a dull one to read." How would you respond to Miss Potter if she had sent you this comment?
Well, I mean, I interact with amateur biologists all the time. And I mean, they're different. Professors perform differently with different students and different professors. But my tendency is always to encourage the citizen scientists as much as possible. And if I'd seen the drawings that she's done, much as I've seen my own students work similar to that, I would encourage it. If for no other reason, then we need to have skilled artists that understand the scientific side of things, making illustrations that others can understand. It's extremely important to deliver the results of science to the public. So I would encourage it. I would have encouraged her.
And my guess is she was being tongue-in-cheek.
Oh, yeah. Yeah.
There is a question here from Andrew Wiltshire. Is the fungi penicillium the same as the penicillin spore that Fleming discovered?
Yes, I mean, the mold penicillium, it actually means in Latin, I believe, little paintbrush. And if you recall, her illustration very nicely shows the spores and chains coming off the end of the hyphae there. So that is the genus of, there are lots and lots of species of penicillium. Only some of them, well, I think they may all produce antibiotics, but only a few of them were the ones that Fleming looked at and developed as antibiotic sources.
There's a question from Ray Heaton. Why do you think she found fungi so interesting to draw as opposed to flowering plants?
Well, she did illustrate flowering plants, but early on she expressed her love of the form and color of the fungi, and I think she approached it first because they were interesting things to draw and paint. And this is one reason why I'm surprised she didn't spend more effort on lichens. Lichens are extremely difficult to draw. If she'd spent time drawing them and illustrating them in detail, would have done a wonderful job. But for some reason, they didn't tickle her like the mushrooms did. Maybe like all of us who collect mushrooms, we're thrilled by the hunt and the fact that they come up all of a sudden and they're there one day and not another. So the Easter egg hunt aspect of looking for mushrooms may have intrigued her.
Yes, and I'm not sure I have this right, but I think in Fairy Caravan, which is after her little, her most famous little books, I think that she kind of gives them a nod like fairies.
Yeah, she makes comments about fairy rings in the woods, and another interesting aspect that it lends itself to fantastical stories, and that obviously appealed to her.
Tessa Graswitz sounds like she is, like Beatrix Potter, a botanical painter. She wrote just a comment, interesting to see the pictures of Amanita rubescens with large chunks missing. They seem to be highly attractive to slugs. I've been painting some recently and the slugs were devouring them as fast as I could work.
Yeah, when you collect wild mushrooms, there's always indication of other organisms interacting with them. She didn't put the slugs on there. They would have been a little hard to do, I think, but definitely she painted what she saw.
Paula Winchester wonders, do you think we would have her marvelous little books if the Linnean Society had accepted her paper or she had continued to work on that? Again, that's a matter for speculation. How would you answer that, Fred?
Well, yeah, I like many others who have speculated about would she have become a mycologist? Well, I think probably very definitely she would have. She definitely had the inquiring mind. She had the scientific mindset of realizing that good science is based on evidence that you see either in experiment or with your eyes. And she would, under other circumstances, in terms of education and upbringing, could very likely have developed into a mycologist. As people have said, archaeologist as well. So it just would depend. It often depends on the teachers you have.
Yes, and where do you get the encouragement?
Yeah, exactly.
Thank you again, Fred. Many people are adding their thanks into the comments. And with that, I will pass it back to Isabelle to close the event.
Thank you, Marta.
I'm still muted, sorry. Thank you very much, Marta. And thank you especially to Fred for a very fascinating talk. I think I've learned lots, which is great. And thank you for all of you who have come on this joined meeting. It's a real delight to be able to hold this, and I think holding it online means that we've had, like, I think at the highest 290 participants, which is absolutely wonderful. So thank you very much for coming, everyone, and check the Linnean Society website for any future events if you're interested. Thank you, and I will close this meeting. Bye-bye.